joined today by Dr. Andrew Stevens, Chief Scientific Officer at Cleo Diagnostics. Andrew, thanks for your time.
Thanks for having me.
A simple tool, a simple biomarker, but a very significant problem. Can you talk about, first of all, the biomarker that you've developed and why it's so important?
Yeah, so this is a biomarker aimed at better diagnosis of ovarian cancer, essentially. It's a very hard disease to detect. It's a very hard disease to diagnose and differentiate from a non-malignant condition. The biomarker that we discovered is a blood marker called CXCL10. It is a very good marker of the presence of cancer. It's very good at differentiating malignant from non-malignant masses. It's also produced quite early in the lifetime of the tumour, so it's a good marker of early stage disease.
And that's the key, isn't it? If you look at the numbers and the market, if we like to put it that way, there's a lot of deaths through ovarian cancer, both in Australia and globally, because we just don't catch it too soon. And this solves that problem.
Yeah, well, that's right. Ovarian cancer is a high mortality disease, primarily because it's hard to detect. So most cases go undiagnosed until they're at quite a late stage and the cancer's already extensively spread, which obviously makes it very hard to treat. The aim here at Cleo is to bring this marker through as the core for 3 different testing scenarios. One of them is general population screening. So this is like where you do a mammogram for breast cancer, for example, or a pap smear for cervical cancer. We want to introduce regular screening for ovarian cancer to pick up those early cancers where they're treatable. At present, there is no screening test for ovarian cancer. Another test that will come through is a test for recurrent cancer. So we know that once women are diagnosed and treated, generally the cancer will come back at some point. And so we want to be able to pick that up as early as possible. And then Cleo's first product to market is something called a triage test. And that test is all about assisting the clinicians to work out before women have surgery whether something is likely to be benign or malignant, because that process and that knowledge helps dictate the kind of treatment that they will have.
And CXCL10 is the byproduct, if you like, or the outcome of over 10 years of your own work at the Hudson Institute. Can you talk us through that discovery story and how you've got this biomarker to where it is today?
Yeah, so it was somewhat of a serendipitous discovery, I guess. It came from a project looking at immune function in ovarian cancers. Ovarian cancer is what's typically called a cold tumor. So some tumors will grow and have a very strong immune response against them, and you can exploit that in treatment. Ovarian cancer doesn't seem to do that. It's very good at hiding from immune attack. What we found was that these tumors were overproducing this CXCL10 protein, which should promote immune function, but actually it was having no effect. And so when we looked further, and it took several years for technology to catch up with what we were hypothesizing, when we looked further, we found that the CXCL10 is actually slightly modified by these cancers. And it's a mechanism that the tumor uses at a very early point in progression to hide from the immune system and promote its own propagation. So as soon as we saw that, we knew that we had something reasonably special because it only happened in malignancy. It didn't happen in benign disease. And that was really the start of test development.
You've recently expanded the biomarker panel from 5 to 8. What prompted the change? But more importantly, what does it mean for accuracy?
So what prompted the change was actually somewhat opportunistic. So the platform that we use for this first test is something called Ella. It's a high throughput platform for immunoassay deployment. The cartridges that actually run the individual marker tests are, all tests are basically complexed onto one cartridge, and it can take 8. So there was no point in leaving 3 of them empty, essentially. The effect of having those additional markers is not so much about increasing accuracy, but increasing the stability of the signal and the stability of the scoring. So we have a couple of what we would call redundant markers in there, so things that are measuring similar effects on the same biological pathway. It just serves to make sure that we can actually capture all of the information and get a nice stable risk score generated at the end.
And you've now enrolled 514 samples in your clinical trial. You've had two very fruitful meetings with the FDA, pre-submission meetings. What does the regulatory pathway look like for this biomarker?
So we've actually completed trial recruitment now. So the trial is finished. All of the patient blood samples are in the freezer. There are actually over 750 patients ultimately that were enrolled. Um, the regulatory pathway is actually very straightforward. So we've not only finished that trial, we've also de-risked and started the manufacturing process now. We're expecting our first manufactured batches of kits within a few weeks to be delivered to us in Melbourne. And the pathway forwards is really one of prescriptive testing. So there are a series of tests that are mandatory for your FDA submission. They're around what's called analytical validation, which is essentially making sure your test kit works the same way each time, and clinical validation, which includes our clinical trial data and also a series of confounding conditions that we test. As I say, it's quite prescriptive, it's very straightforward, and it's simply a case of measuring samples now and running through quite a substantial number and then assembling the FDA paperwork. So we're expecting to have that completed around the end of the year for submission very early in Q1. And then the approval process is a 90-day turnaround time. So we're working very hard to de-risk any questions that might come back. And hopefully we should have that market approval around the middle of 2027.
The test is initially targeting the pre-surgical triage market in the US. Why this approach, and what does it mean to clinically validating the product?
So the approach really is a strategic one of being able to get a test out into the market in a reasonably short space of time while we're working on some of the bigger ticket items, particularly a screening test. It's also dictated by the size of the clinical trial you need to do. So a trial for this pre-surgical test is relatively small. A screening test, obviously, is extremely large. The market size for that pre-surgical triage, however, is actually pretty substantial. So in the US, there's about 2 million women every year who have an adnexal mass investigation. Now, those are the women that will benefit from a Cleo test to assist in determining whether something is likely to be malignant or not. So that's our first rollout, and it's the simplest market to engage with, and it also gets Cleo's name out there into the market and gives the market some confidence for our next set of testing that will come through.
10 years of science culminating in a simple blood test that will genuinely change the lives of women with ovarian cancer. We genuinely follow it with interest and we look forward to the news flow ahead. Andrew, thanks for your time.
Thanks very much.